Morning online.
Yes, ma'am.
Hello. Yeah.
Yes, ma'am. Good morning.
Good morning. আচ্ছা ব্রেক টাইমিং সবাই ক্লিয়ার না? নিজের থেকে বলবে বা স্যার 5 মিনিট ব্রেক এখন ব্রেক আছে। And the worst part is that today the person rang the bell at 11:43, which should have been 11:40. Okay, so I don't know what the thing, because 5 minutes of break you had. So that is what I got confused.
7:46 से 7 बजे।
हैं?
मैम हमारे आकाश में तो कोई गैप ही नहीं दिखा रहा, लिखा रहा है कि 3 आवर क्लास।
शेड्यूल देखो मेरे पास है।
एक ही दिन बचा है।
क्लास का क्लास कर रहे
नहीं नहीं नहीं, बीच में 5-5 मिनट का ब्रेक है। तुम लोग 4 मिनट।
अरे आगे बच्चे खाली
This is the timing. See? देख लिया?
ছবি से तो
अरे আমি দেখি কি করব, ওরাও তো দেখছে সেম শিডিউল।
मैम
ওটা জানে না নাকি?
पता ही नहीं है। तो ऑनलाइन एक देखिए
স্যারও সেম গ্রুপেই আছে, সেম গ্রুপে গিয়ে টাইম টেবিলটা দেখছে। দেখতে হবে টাইম। 225 आज मिनट एक्स्ट्रा पढ़ा दिया। वाह वाह वाह। না না, আমি তোদেরকে 15 মিনিট 215 ছেড়ে দেবো, আমি পড়াতে পারবো না।
না এটা তো বড় যার এতক্ষণ ধরে বসে আছে।
1-5 don't get shocked. Okay. সত্যি যাচ্ছে তাই অবস্থা।
দাম পজিটিভ
একদম positively আটকে আছি। Listen, that 1 hour of class where বারে বারে আমি সেই prokaryotic cell নিয়েছি, okay, that messed up my entire schedule. Okay, now we would have completed prokaryote, আজ cell wall শুরু হতো। Okay, because এখন যে এটা আচ্ছা cell wall we have done in prokaryotic cells. Chromatophores was the last thing we wrote. Okay, next we come to the next structure is the surface structures. Okay. Okay, so first let us talk about surface structure. So we have finished with lysosomes and chromatophore surface structures. Now bacteria can be, bacteria has two types of surface structures. One is the motile structures and another is the non-motile structures. So with motile structures, because some bacteria are, have show movement, so they have flagella. And the other non-motile bacteria, they just flow through the fluids or something. They do not have such movements. So they require external kind of things for movement. Okay. So coming to surface structure, there are two types. Okay, based on motility, or attachment. Based on motility or attachment. Attachment of bacteria. Okay, because one is flagella. So the first surface structure is flagella. Okay, so bacteria may be motile or non-motile. If it is motile, then they have this particular flagella made up of flagellin protein. Bacteria may be motile or non-motile. Motile or non-motile. Okay, motile form of bacteria. Okay, contain flagella. Contain flagella. So NCRT has not given any diagram of the flagella, but your module has given one particular diagram showing 4 rings and 1 hook and 1 filament. Okay, so motile form of flagella. So what is the flagella basically consists of? So flagella consists of 3 parts. Okay, so I'm going to do a slight diagram here so that you understand what are the 3 parts and how does it affect. So suppose this is the plasma membrane. And this will be peptidoglycan layer. So you have one ring here and another ring here. And these two are connected. And then there is a hook-like structure. And then we have a filament. Okay. So this is the prokaryotic flagella. Which prokaryotic flagella? This is gram positive, not gram negative. Okay, gram positive, gram negative diagram is given in your module with the 4 rings. Okay, LPSM it is known as. L means lipopolysaccharide, S means LP means peptidoglycan, S means supramembranous, supra means above the membrane, and M means membranous, meaning within the membrane. So there are 4 layers to that basal body. Okay, and then we have the hook that is coming out of the surface, this peptidoglycan layer, and then the filament is the longest portion of the flagella that comes out of the cell surface. So that is what it is written for the filament. Now the filament is made up of flagellin protein. This entire structure is not made up of flagellin protein. Only this part is made of flagellin protein. Hook has some different type of proteins, and basal rings are of different types of proteins. Now this is not like the eukaryotic basal body. This kind of rings do not have that 9+0 organization, or neither they have originated from centriole. Okay, so they are completely solid structures. Okay, like in centriole basal body, they have a hollow structure where the central position is called hub, and you have 27 microtubules in the periphery, just similar to centriole organization. But that is not seen in prokaryotic flagella. And the arrangement of this flagella is also not 9+2. Okay, this arrangement is mostly helical. Okay, so the flagellin protein is arranged in helical. But this is like in NCRT, the only thing they have mentioned is that this is like called as the basal body. Okay, they have just mentioned basal body for the flagella, nothing related to this basal body. We can write this ring like. Okay, so this is the only thing that is mentioned about this. Then you have a hook. Okay, that is like curved structure. Now hook basically attaches the filament to the basal body. Okay, so hook's function is to attach the filament to basal body. Okay, so this is the thing, and finally we have this structure which is called as the filament. Okay, made up of flagellin protein. Flagellin protein, longest portion of flagella, extends out of the cell surface. Extends out of the cell surface. Okay, so these are the 3 points in relation to the filament.
मैम ग्रीनটা কি প্লাজমা মেমব্রেন না সেল ওয়াল
Green is plasma membrane.
আর
Wait, I will,
ব্রাউনটা সেল ওয়াল
brown na, the yellow.
মানে ওই হলো আর কি
মানে color blind হয়ে গেছিস কেন? This is called as the M, মানে membranous. Okay, this is the membranous layer. Okay, this structure is called M, and then above this portion is called supramembranous. So this layer is called S. মানে supra মানে above the membrane. Okay, this here is your peptidoglycan. Okay, so from the peptidoglycan layer, the hook is emerging outside. If the hook is not emerging outside, then the filament will be stuck here, and the filament will not come out of the surface structure. Okay, then the movement will also be hampered. They won't be able to move. Okay, so this is the thing. And if you have another layer above it, which is a lipopolysaccharide layer, then we call it as L. So in your module, LPSM is given like that. But in NCRT, they have not mentioned anything about this. They've only mentioned that 3 parts are there: basal body, hook, and filament. Filament is the longest portion, which comes or extends out of the cell surface. That is it written about it. And obviously, it's made of flagellin protein. Flagellin protein tau can I mention? NCRT is not mentioned that prokaryotic flagella is made of flagellin protein.
ম্যাম ওই ফ্ল্যাজেলাটা কি করে স্পিন করে
Basal body এরকম rotate করবে, এখানে rotator motor আছে, so they take ATP here. ATP hydrolysis or basal body rotates, the hook will rotate and the filament will rotate like this. Filament rotation এরকম হবে.
মানে অ্যাকচুয়ালি মানে ক্লকওয়াইজ
Clockwise, anti-clockwise, depending upon it is going forward or backwards.
Basal body is also protein.
And why not? হ্যাঁ,
ম্যাম এস রিং এর ফুল ফর্মটা আরেকবার বলুন
কোনটা?
ওই
LPSM.
এম এস যেটা আছে হ্যাঁ হ্যাঁ
S basically means supramembranous. Supramembranous means above the membrane. One basal ring is in the membrane and one basal ring is above the membrane. So this is called supramembranous basal ring. So S represent that part. So in your module you have LPSM. So P will represent peptidoglycan, and if there was this kind of like a lipopolysaccharide layer here, then it would have been called as lipopolysaccharide ring. Okay, and then
Okay,
there'll be
thank
4 layers like this, 4 rings like this, then the hook comes out. Okay, so but it is not like it's between this in the middle of the membrane and then just above the membrane.
4 rings.
Now 4 rings because I'm talking about supramembranous. 4 ring structures there in your module.
A2 ring.
It's a gram positive bacteria. In your module, they have given gram negative bacteria. But in NCRT, nothing of this structure is mentioned. Okay, the diagram is also not given of flagella in NCRT. Okay, just to explain you how does it look in your module, it is given. Okay, and nobody will ask you related to what is LPSM and what is SN. They're only going to ask you what is the filament made up of. What is prokaryotic flagella? What kind of protein is it made up of? Eukaryotic flagella, what kind of protein is it made up of? Whether prokaryotic flagella shows 9+2 organization or not. That's the only questions that are going to be asked in this. Okay. So coming to this, after this flagella part, we can write one more thing. Bacterial flagella may vary in number and arrangement, but they have not mentioned how many in number or the types, in number and arrangement. So you can have monotrichous, you can have amphitrichous, you can have lophotrichous. Okay, so monotrichous means a single flagella. Lophotrichous can mean 2 flagella coming from 2 sides. Lophotrichous means a tuft of flagella coming from one side. Tuft, মানে multiple, yes.
আমি এখানে 9+2
যখন সিলিয়া ফ্ল্যাজেলা ইউক্যারিওটিকে পড়াবো তখন বোঝাবো। So here bacterial flagella vary in number and arrangement. So this line is mentioned, but without any examples. Okay. So lophotrichus, amphitrichus, monotrichus, peritrichus, atrichus. There are 5 types. Okay. Atrichus means no flagella. So they are non-motile bacteria. Peritrichus means flagella is surrounding the entire cell. Peri means surrounding. Trichus comes from the word trichome, which means hair. So flagella is technically hair-like outgrowth. Okay. Now, if we talk about lophotrichus, tuft of flagella from one end, amphitrichus, 2 flagella at each end, or a tuft of flagella at each end. That way. So these are the different types based on arrangement. And number also, you can say monotrichus, ditrichus. Okay, ditrichus, which is again amphitrichus based on 2 corners or 2 poles. We can say it like that. But it is not very clearly distinguished in NCERT. They have just written the statement like this. Okay. So this is about your prokaryotic flagella. Next, what we're going to study about is the non-motile or the non-locomotory structures, which is basically pili and fimbriae. Okay. So you have non-locomotory structures, which comes under the surface structures. They are pili and fimbriae. First, let us write about pili, which is obviously plural, and singular form will be pilus. Singular. Okay. Pili, short tubular structures. Okay. Present as a cell surface structure. Pili, the function of pili is not given. It is just mentioned it is made up of specialized protein. That protein is mentioned in your module as pilin protein. Okay. Apart from that, nothing in the NCERT has been mentioned. Pilin protein is also not mentioned. Module mentioned kara ache. And one more thing that is not mentioned in about pili is that pili helps in the attachment of bacteria during conjugation process. The F plus F minus conjugation, HFRF minus conjugation. These are different types of, you can say kind of representing sexual reproduction, but not technically sexual reproduction. Representing how? Because a DNA material has been transferred from one bacteria to another. Kind of like a genetic material being transferred. So that's why we say in sexual reproduction, that is what happens in the offsprings, right? You transfer a genetic material from the parents. Yeah. So it's not true sexual reproduction. It's like a falsified version of it. So in that you require pili, because pili of both the bacteria are going to fuse together and form a conjugation tube. এখানে না cilia কোথায় আছে? Prokaryote does not have a cilia, একদম না. Okay, cilia is a eukaryotic thing. Okay, and ore-like working because of the synchronous metachronous movement, like this. That's why. Okay, when you want to go forward, they go like this. Okay, when they have to go forward, backward, they go like this. On a backstroke or front stroke. Swimming, it's a swim, right? So the front stroke is like this, and backstroke is like this. That's what cilia does. Okay, so this is what it is. Pores. Okay. নৌকোতে বস, নৌকোতে লোকটা যখন চালাবে এরকম ভাবে সামনে যাবি, এরকম ভাবে যখন চালাবে পেছনে। That is how cilia works. That's why it's called pores. The pili is short tubular structures. Short tubular structures. Okay. Made up of a special protein. Made up of specialized proteins. This much only is written in NCERT. Okay, but what is written in module? That is they have added here, and they have mentioned pilin. So we have these kind of proteins found in prokaryotes, which we can't see in eukaryote. You will not see pilin protein in eukaryote. You will not see fimbriae, which is fimbriae made up of in eukaryotes. Flagellin, pilin, fimbriae, all these types of proteins, you don't find it in eukaryotes. Okay, so this was about pili. The second one is basically fimbriae. Fimbriae, they have mentioned that it is bristle-like structure. Bristle-like structure means the toothbrush. The brush part, which is like pointy and very thin sort of, that is called as a bristle. So bristle-like structure, okay, extends out of the cell surface, Now, function of fimbriae is very clearly mentioned in NCERT. First function is attaches the bacterium to the rocky surfaces in streams. Okay. Second function is attaches the bacteria to the host tissue. Okay. So it is basically attachment. This is for attaching the bacteria to bacterium together. Okay. This is for attaching the bacteria to other surfaces. Okay. So attaches the bacteria to rocks in streams, okay, as well as host tissues, So how is the question going to come if by any chance it is asked in NEET? They're going to ask you short tubular structures made up of specialized proteins. That's one question in prokaryotes. And they're going to give you options. Okay, pili, fimbriae, flagella, cilia. So you know the answer is pili because the term is short tubular and made up of specialized proteins in prokaryotes. And in fimbriae, bristle-like structure is one thing they're going to point out. Bristle-like structure that attaches the bacterium to rocks in streams, or they can say attaches the bacterium to host tissues. So that would be your fimbriae. So these are the pointers that you need to know. This one and this bristle-like structure. Only questions that is going to come in it. If they find chalo, let us give the students a very easy question. They at home they're very happy. No fight with the wife. So set up a question which is easy, makes the students happy. আর যদি বাড়িতে খুব ঝগড়া হয় বাম্বু খেয়েছে বউ থেকে তাহলে রাজ্যের assertion reason or statements. So 2022 paper and 23 paper. Okay, 23 paper was pretty easy. 2022 paper এ রাজ্যের statement and assertion reason দিয়েছিল একদম anatomy দিয়ে এবারে পুরো চলে এদেরকে পাগল করে দিয়ে anatomy. So they gave so many questions out of
চারটে না পাঁচটা ছটা করে দিয়ে দিয়েছে
so you can understand mood এর উপরে depend করেছিল. Okay, দিয়ে ঝগড়া করে এসেছে দিয়ে দিয়েছে. It's kind of like correcting papers in school.
কারোর কাছে নিজের ডিজাইন গুলো বলতে পারতো না তাই কোশ্চেন দিয়ে দিয়েছে
No, you বলছে কি বললি আবার রিপিট কর. What did you say?
বললাম এক্সামিনার কারোর কাছে ডিজাইন গুলো বলতে পারতো না তাই কোশ্চেন দিয়ে দিয়েছে
ঠিক আছে, very good. So technically these are very simple questions that can come from prokaryote. The prokaryotic cell maximum আসলে একটা question, just one question. Okay, so what can be that kind of a question? That is like cell chapter, so only 2 questions this year came. Okay, one was mark the following and one was mitochondria, nothing else came. That too eukaryotic cell. Prokaryotic cell to gone. Okay, 3 pages of prokaryotic cell you studied and nothing came. So emphasis like, see, it did not come this year, does not mean in next year they're not going to ask me from prokaryotic cell. So you have to go this way. Okay, worst case scenario দিতে হবে কি আসতেও পারে, like morphology সবাই পড়ে নিয়েছে তারপর দেখতে no families. 3 questions came. Yeah, about that seed and about that perigynous and everything, but nothing, no families. So that is the way it's like prepared. Okay, so we are done with surface structures, we are done with the cell envelope and whatever extensions of the cell envelope. Now we are going to come a little bit inside. So the matrix is known as cytoplasm. Okay, fluid filled matrix. Fluid filled matrix known as cytoplasm. Cytoplasm is present, but technically in prokaryotic cells, cytoplasm is the only place where all the cellular activities takes place. Minus the lives, the mesosomes where respiration is taking place or in aerobic bacteria. Cytoplasm is showing glycolysis. Cytoplasm is showing replication of DNA. Cytoplasm is showing transcription, translation, all these processes. So we can also use that statement. Many, the metabolic activities occur in the cytoplasm that render the cell in a living state. Right. So we can use that term here also as an assertion reason question. Okay. So in the cytoplasm, what all are present? So after the cell surface structures, you've got the, if we have cell organelles anytime present, so what do you have? No membrane bound cell organelles. No membrane bound cell organelles. No membrane bound cell organelles. Okay, this is absent. The only cell organelle that is present, the only non-membranous cell organelle that is present is ribosome. Okay, the only non-membranous cell organelle present is 70S type of ribosomes. 70S type of ribosomes. Okay, so this is about the cellular organelles. Board has its own AI, I feel. Okay, so non-membrane cell organelles except for ribosomes. What I would say.
ম্যাম বোর্ডটাও টায়ার্ড হয়ে গেছে।
এই তো সব শুরু হলো রে।
এই সবে শুরু হলো রে, এখনো সেল চলছে।
সবাই থেকে বসে
till biodiversity and conservation, তারপর দেখবো এক দুজন আসছে এখানে, বাকি সব কটা অনলাইন। ঠিক আছে, বায়োডাইভার্সিটি কনজারভেশন অব্দি আমি দেখছি পুরো গোটা ক্লাস খালি। I am teaching the walls.
বডি পুরি দেওয়া
হ্যাঁ,
বডি পুরি দেবে
বডি তো পড়াবো, কোথায় পড়াবো? ফাইন ল্যাঙ্গুয়েজ করবো নাকি?
হচ্ছে পিডিএফ থেকে ভিডিও পড়ায় অনেক টিচার
পিডিএফ দিয়ে না, আমার পিডিএফ ভালো লাগে না আনটিল আনলেস মানে খুবই গ্রেভ সিচুয়েশন। ওকে, লাইক দেয়ার ইজ নট মাচ টাইম লেফট, then I will teach in PDF. Otherwise, biodiversity conservation, mostly it is informative, but there are certain places like species area relationship today मैं पढ़ाते शुरू करी PDF এ। ওকে, কেউ মাথার উপর দিয়ে এরকম করে মানে একটা ওদের অর্গানে কি রেক্ট্যাঙ্গুলার হাইপারবোলা আছে, তোদেরও যাবে ওরকম। ওকে, সো রেক্ট্যাঙ্গুলার হাইপারবোলা বলে তো, is it correct?
Yes.
না 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 না, the graph is correct, তো rectangular hyperbola
চলে গেছে।
হ্যাঁ বুঝে, আমি তো নিজেই খুঁজছি পেছনে এসছে।
Yes, ma'am.
চলো, কি মানে I don't understand what it is. Why is it stuck? Somebody else is writing on the board or what?
স্যাম ডিসপ্লেটা মানে ডিসপ্লে করে দিই
আবার restart in every solution, মানে problem there's a modern solution restart. Online please just wait, I think the both gone. Restart ও চলবে না, direct off করো। Okay. Hello everyone, I'm back. Sorry. 10 minutes more I'll teach and then you can take a break till 1:15. That's a break is till 1:15 by the way. Just 5 minutes before 1:20. Like, I don't know what's so nice about it. And I was under the impression, the break is from 1:20 to 1:50. Okay, so
ম্যাম ভালোই করেছে বাস আমার 20 তে আসে আর তারপরে 50 তে আসে
yeah. What's a bus?
इसलिए 15 या 50 का निकल जाएंगे
Okay. Chalo, so this is about the cell organelles. Now we come to the genetic material. Okay. So there's like a prokaryotic cell. Okay, prokaryotes consists of a single double-stranded circular. Okay, single double-stranded. Single ही होगा ना, single chromosome we are talking about. Single double-stranded circular DNA. Single double-stranded circular DNA. Single because a single DNA is present in the cell. Okay, so that is why it is called as that. And single chromosome because the DNA is not made into parts and placed into different types of chromosomes, like 23 pairs we say, or like a genome set we say, the 23 set in case of humans. So our DNA is 6.6 into 10 power 6 base pairs. So the 6.6 10 power 6 base pairs of DNA is actually divided in 23 pairs. And each pair has some amount of the DNA. So that way the 2.2 meters length is reduced to that. Okay, to a 700 nanometer thread. Coiling, super coiling. So that is the way our DNA is being. But for them, they do not have that much kind of like a coiled structure, like too much of super coilings and all and embedded in separate chromosome. They have a single chromosome, we call it, because they have a single structure, single DNA coiled by polyamines and present in the cell. Okay, because there's no nucleus. The size of a nucleus is 1 micron. And you're reducing a 2.2 meters length of DNA into a 700 nanometer width of thread. So it is very, very short. Okay, so that is the way we do it. So single double-stranded circular DNA, okay, which is the genome, or I can say genetic material, genome or genetic material. Also known as a single chromosome. Also known as single chromosome. Now, this particular DNA is found in a region in the cytoplasm called as nucleoid. And and obviously this DNA is attached to the mesosomes. Okay, so nucleoid is not mentioned in prokaryotic cell NCERT, but we will write it here because later on we do come across it. Okay, so DNA is present in a region of the cytoplasm called as nucleoid. Cytoplasm called as nucleoid. Called as nucleoid. Okay. So nucleoid means nucleus-like. DNA is naked because there is no nuclear membrane surrounding. So DNA is naked. DNA is naked with no nuclear membrane surrounding it. Or we can say as there is no well-defined nucleus, we can say. As there is no well-defined nucleus. So this can be stated as an assertion, reason, question altogether. Okay, prokaryotic DNA is naked. Suppose that's the assertion. Reason can be no well-defined nucleus or nuclear membrane is absent, okay, or the DNA is not enclosed in chromatin reticulum. Any of these three statements will become the reason. Okay, so this is an assertion reason statement from here. So after this DNA, this regulates, controls every activity of the cell. Okay, then we have something called as an extra chromosomal DNA.
See, correct explanation to one
Key.
the DNA is naked and as there is no well-defined nucleus.
Yeah.
Explanation
কেন হবে না? তাহলে prokaryotic বলি কেন? Naked DNA মানে কনসেপ্টটা কি? If the DNA, আচ্ছা eukaryotic cell we don't say naked DNA, right? So when we talk about no well-defined nucleus, we are technically talking about nuclear membrane absent, chromatin reticulum absent, nucleolus absent, nucleoplasm absent. Well-defined money, you are not defining the nucleus there. Like a double membrane, presence of double membrane, nuclear core, nucleolus, nucleoplasm, chromatin reticulum. This is called defined definition of a nucleus. So if that is absent, that means DNA is naked. Okay, so this is how we state the assertion. Now coming to the next thing. Okay, many prokaryotic cells or many prokaryotes consist of extra chromosomal DNA molecules. Consist of extra chromosomal DNA molecules. DNA molecules. Okay, known as plasmids. Now, plasmids is not a genomic material or a genetic material. Okay, plasmid is an extra DNA that gives certain unique phenotypic character. Like one is antibiotic resistance, and later on you study it helps in bacterial transformation process using foreign gene. So that's basically your biotechnology part. Okay, so plasmids are also circular double-stranded, by the way. Okay, plasmids are also circular double-stranded. And gives unique phenotypic characteristics. Characters to the bacteria. Okay. Here, example would be antibiotic resistance. If the question comes that certain bacteria are resistant to antibiotics and the reason is that plasmids are present in such bacteria, that will be the correct thing. So this can be stated as an assertion reason question. Okay, so this can be assertion, this can be the reason. Okay, what if we change the assertion reason? What if this is the reason and that's the assertion? गलत कहा हुआ है, सही आंसर है लेकिन सेकंड ऑप्शन ही होगा कि वन ही हो सकता है. If I say कि चलो plasmids give unique phenotypic characters to bacteria, reason is plasmids give antibiotic resistance to the bacteria. It's like a palindrome. अब किसी को भी इधर assertion बनाओ, किसी को भी reason बनाओ, वो आंसर वन ही दे. ठीक है, तो इसको ऐसे समझना है कि अब पहले assertion reason देखो कि इसको ये इसको explain कर रहा है कि नहीं. अगर कर लिया तो ऑप्शन वन है, अगर नहीं कर पा रहा है, बहुत ही अब्रप्ट लग रहा है, दोनों स्टेटमेंट करेक्ट है लेकिन कर ही नहीं पा रहा एक्सप्लेनेशन, तब वो ऑप्शन टू होगा. ओके, द रेस्ट कम्स लेटर. ओके, सो यू हैव टू ऑलवेज चेंज देम, इंटरचेंज देम. एक बार असर्शन में इसको रीजन बनाओ. ठीक है, सेल मेम्ब्रेन में भी ऐसा ही है. Gassy fluid nature enables the lateral movement of protein. Okay, this ability to move between the membrane is the measurement of its fluidity. That's your assertion reason there. You can make two assertion reason question from it. Okay. So this is the way you can actually, you have to think about this कि इसको कैसे हम लोग इधर से कितने types of assertion reason आ सकता है. Okay, so this is actually correct. इसको आप interchange भी कर दो, option 1 देगा. Okay, so this is about plasmid. और एक है कि plasmid helps in bacterial transformation process using foreign gene. Plasmids helps in bacterial transformation process using foreign gene. अच्छा, अब मैंने अगर इसको दिया और इसको दिया, इसको assertion दिया और इसको reason दिया, करेक्ट होगा? करेक्ट तो है. What will be the option? 2 will be the option. क्योंकि वो explain नहीं कर रहा है. Unique phenotypic character तो antibiotic resistance. Phenotype मतलब क्या? Your blood group is also a phenotype. It's not, genotype is when you are actually finding whether you are heterozygous or homozygous. But what kind of blood group you have, that is a phenotype. तो ऐसे ही अगर पकड़ लो तो antibiotic resistance a phenotypic character. Okay. तो उसके लिए इसका जीनोटाइप होना चाहिए कि उसमें उस एंटीबायोटिक के अगेंस्ट काम करने वाला मेडिसिन केमिकल होना चाहिए. That's the genotype. Okay, so this will be option 1, but if you give this and this as the option, it is both are correct, but R is not explaining the assertion. This can also be used as a reason. ये भी एक reason हो सकता है. इधर तो ये reason है तो ये assertion भी हो सकता है, इधर assertion है तो ये reason भी हो सकता है. लेकिन यहाँ पे ये reason आएगा इसके लिए. अगर ये दोनों अलग अलग, ये plasmid जो antibiotic resistance, plasmid helps in bacterial transformation, वो भी option 2 ही है. No, they are not related to each other in any way. Okay, they are the two statements coming for plasmids. So this is the way assertion reason will come. Okay, so from here we can make at least 3 assertion reason questions just by these 3 statements. 3 are there. Okay, so this is about plasmids. Next we have please. बचता क्यों नहीं है भाई? चलो, take a break for now. Keep it in a logged in state. Yes. অনেক কটা ডাউট আছে. এরা কাজ করে এখন খেয়ে নে, লাঞ্চ নে, ব্রেক তো আবার সেটা আমি আসবো, বলবি কি ম্যাম বেটা খাইনি. তো অনেক কটা ডাউট আছে, তোকে দুটো ফোনের পরে বাস ধরতে হবে. ভগবান, আয় তাহলে চলো, টেক আ ব্রেক. হ্যাঁ, অনলাইন I'll take your doubts right now. There's a 30 minutes break. Please go and have lunch.
আমি তো কি করবো কারণ
আমি দেবো এই সায়নী
ওই ঢাকি না দরকার খেয়েছি Could you please send me the
এটাই খাবো
না ছেলে আমি পিছনে না না এটা কি বাড়ি গোল্ডেন জিনিস বেশি মনে তার জন্য ওকে একটা ডিসটেন্স নিয়ে তারপরে
এই যাবি ভাই
সেট করে দাও কি করে কিডন্যাপ করবে এখানে না তো ট্রাফিকিং করে দেবো এই বাবা মানে করে ব্ল্যাক চেক দিয়ে দেবে
আমি উঠবো তো
আমি দিলাম না তো মামার সাথে যাচ্ছে এখন ছিলাম কিছুদিনের জন্য বাবা exam PIT is approved. Yes. Major top party is one stage. Yes. বাড়িতে ছিলাম আরেকটা ছিল দক্ষিণ বলেছে আমরা দুজনে মানে বাগান তখন
তাই আমি এখানে
তখন বুঝে যাচ্ছি
কি বলেছে মানে আগে টিএফএ ছিল না সিএফএ ছিলাম নিশ্চিত কোয়ালিটি ছিল সেটা অ্যাডমিশন দেয় তারপরে পড়লো
ফার্স্ট ল্যাঙ্গুয়েজ সেটা ট্রাই করো এগুলো অনেক কারণে কোন ল্যাঙ্গুয়েজ নিয়ে প্রবলেম হয় না বিষয় একটা পিসি অ্যাডভান্টেজ
অনলাইন
না
Actually, acha, it's a friend you don't know.
Yeah.
They are asking for violent. I think the Botany,
Yeah.
okay. ওরা বাঙালি ওটা তো most like লোক Botany.
নাম্বার
By the way,
आगे बढ़ रही है 500 प्लस যেকোনো একটা চিজ করে চিনতে পারে
তোরা তোরা কি ফেভিকল লাগিয়ে বসেছিস যাবে তোদের মুভিটা দেখলাম না সিরিয়াসলি একটু ওঠ হাঁটা চলা কর ভাই কি বসে বসে তো যাবো মানে বসে বসে ব্যাটারি চার্জ করার মতো দাঁড়িয়ে বসে আছে
আমি কেন হ্যাঁ এটা বুঝতে পারলাম
चलो आ जाओ ऑनलाइन गुड आफ्टरनून
Good afternoon, ma'am.
Good afternoon. Acha, just wait. I think you have to join the second link. Online, please join the second link, okay? There are two links given for the
Okay,
same
ma'am.
class. Okay. Chalo. Any doubts till prokaryotic? is release initiation factors, the ones like the elongation factors, TFTS, TFTU for eukaryotes, or you have this TFG translocase thing. So these are the initiation, these are the elongation factors, initiation factors. So all these things are involved. Release factors also. Once the RNA, the A site reaches reaches a stop codon. So then it signals release factor. So ribosome has a lot of role to play. It has the sites for tRNA to enter and to tRNA to exit, and the peptidal site for the peptide bond formation. So wherever the peptidal site is, there you will have 23S rRNA. So it is on the largest subunit that you see this kind of formation, peptide bond formation. So this is what exactly prokaryotic ribosome is. So you study all this in molecular basis, the detailing of it. In this cell chapter, they have not explained much about it. Okay, just the size, where it is associated mostly, that is plasma membrane, and what is polysome. These three questions will come, and subunit wise 50S and 30S. Okay, again we study ribosome as pallade particles, or George Pallade found it in 1953. Given the date is given in NCERT. Okay, so there you will study about this 80S ribosome. Okay, and 70S we have already studied. So mitochondrial, chloroplast, organella ribosomes. There are 4 organs or organelles where ribosomes are attached. Nuclear membrane, ER, mitochondria, chloroplast. Attached or present. So this is the thing. Next we come to the next part, inclusion bodies. Okay. Last part of prokaryotes. So we have started from an envelope. We spoke about the cytoplasm, the cell surface structures, locomotory structures, DNA, plasmid. Okay, now we come to inclusion bodies. So these include reserve food material, include reserve food material, as well as gas vacuums. Okay. Now, in terms of reserve food, what are the reserve food material that we see in inclusion bodies? Reserve food material, these are the ones that do not have any membrane surrounding them. They are floating in the cytoplasm or lie in the cytoplasm according to NCERT line. Okay, so do not have a membrane. Do not have a membrane surrounding them. Membrane surrounding them. Freely lie in the cytoplasm. Freely lie in the cytoplasm. That is basically what we talk about the reserve food material. Examples, you have phosphate granules. Yeah, phosphate granules. You've got glycogen granules. And you have cyanophycean granules, which is technically blue green algae starch. Yeah, cyanophycean starch we call it. So cyanophycean granules, cyanophycean starch we can say. So, etc. You can have a lot of things. Phosphate here. Phosphate granules, glycogen granules. Glycogen granules are generally heterotrophic mode of bacteria. Those are heterotrophic mode, saprophytic bacteria or any kind of parasitic bacteria, they basically store glycogen. Phosphate granules are stored by, like, they are actually called volutin. These granules are called volutin granules. They're stored by those chemoautotrophs. Chemoautotroph or chemoheterotroph. Those who require the phosphate mineral, or they take phosphate type of mineral things and they store it in them. So that's like a reserve food for them. And cyanophycean, we know it's cyanophycean starch that is produced during photosynthesis. And you have other also, ferritin granule is also present. Iron bacteria, ferrobacillus, chemoautotrophs and chemoheterotrophs, they have these different types of mineral ion granules, which is as the stored material. Okay, So this is about reserved food material. They do not take part in any kind of metabolism. They are the products of the metabolism. Okay. Next, we have gas vacuoles. Now, NCRT prokaryotic cell has not mentioned much about gas vacuoles. They have said that it is found in cyanobacteria, green bacteria, and purple bacteria. Okay, so these are vacuoles, or these are found in cyanobacteria, blue-green algae, yeah, green and purple bacteria, But apart from this, they have not mentioned anything about it. At least in NCRT, module has given a term that gas vacuoles do not have a tonoplast. They do have a protein vesicle. They are a protein vesicle, but that is not a tonoplast. So they are called as pseudovacuoles, and their function is to store gases like carbon dioxide, sulfur dioxide, and all, as well, and it helps in floating or buoyancy. So that is written in module. But again, in NEET exam, there are not much questions related to what it stores and all. That has come. AIPMT, yes, there's a lot of questions related to vacuoles, but NEET, at least for this after 2016, there is nothing, no mention of gas vacuoles as such. So this is the only thing that we need to know according to NCRT. If you go into the module, we can say it is known as pseudovacuole module. Later it is written pseudo does not have tonoplast. Okay, so that is why it is called as unit membraneless. I mean, it does not have a unit membrane. Okay, it does not have tonoplast, known as pseudovacuole. Because it gives the appearance of a vacuole, because it is storing things, but not that it takes things against concentration gradient and stores inside. Okay, so functionality stores gases and provides buoyancy. So this is all about your prokaryotic cells, starting from every structure, from the cellular envelope to all the general description. This is all. Okay. So after this prokaryotic and prokaryotic cell description, we have eukaryotic cells. But before we start that, we'll write certain differences between prokaryotes and eukaryotes. Then we go for eukaryotic cell. Okay. Clear about this?
Yes, please.
Okay. No doubts in prokaryotic cell? All clear? Okay. Chalo. Difference between prokaryotes and eukaryotes. ওটা করে দিয়েছি তোমরা তো এমন ভাবে বলছিস হাঁ করে বলে আবার লিখতে শুরু করে দিয়েছি বলবি তো আমি করে দিয়েছি Okay, done. So difference between prokaryotes. Okay, then we find. We'll start off with eukaryotes. NCRT, whatever has written, right? Eukaryotic cell. Hello, finally we started. Eukaryotic cell. Found in or includes. What do these cells includes? Protistans. Yes. Fungi, plants, and animals. Fungi, plants, and animals. Okay. Now, eukaryotic cell shows high compartmentalization. Eukaryotic cells show high compartmentalization. Compartmentalization internally. Internally. Is your assertion. Reason. membrane bound cell organelles are present in eukaryotic cells. First option. Your second line statement itself is an assertion reason. Okay, so membrane bound cell organelles, membrane bound cell organelles are found. So this is the first assertion reason. What if I change the reason to assertion and the assertion to reason? The same thing it will come. Because either or the way they are explaining each other. And option one only. Okay. So always remember when I'm doing this now, you try to change the statements and see whether they in both ways they are coming as option one or not. Then you will understand how you have to answer assertion reason. An NCRT and NEET exam, NTA will always focus on NCRT statements for assertion reason. Or if they don't, they will just twist that. When you just change the the structure of the statement, meaning will be same, the structure will change. Okay, so the concept you have to grasp. When you're talking about high compartmentalization, we are talking about membrane-bound cell organelles. If I become more specific, that eukaryotic cells, show high compartmentalization. ER has extends into extraluminal and luminal space. Divides the cell into extraluminal and luminal sites. That is also statement one. Answer will be one. Because this compartmentalization is because of ER also. It stretches from the nuclear membrane till the plasma membrane. Okay, so that kind of creates those cytoplasmic things and also the internal ER lumen. Okay, so this was about the next. The next point is eukaryotic cells obviously vary in shape and size. Okay, which also determines their function. So eukaryotic cells vary in shape and size. Shape and size will determine their functions. মানে বেশি কি চালাচ্ছি না না ঠিক আছে গরম লাগছে আমি দাঁড়িয়ে দাঁড়িয়ে ঘুরছি তো আমার গরম লাগছে আরে ওকে I have my phone, I have MI remote, I'll switch it off. Okay, if anybody is tall enough, please close this flap. If you can close that flap, then it will be easier. Who's tall? Tallest person here. Yeah, just don't make any noise, otherwise people will run here like what you're doing. Yeah, you can just close that. হ্যাঁ খাতা দিয়ে better হবে না which side this side you want that close or this side আর ওর এটা দরকার আছে রুফের মাথা আটকাবে আরে don't do it parallelly, do perpendicular that board. And that's called perpendicular. Yes. You do it with the corner. No, I'm not switch it off because it's literally I'm feeling hot right now. Like proud brother. সে ভাই বলে দিল এই জন্য কাজ করতে নেই
এরপরে হুট করে বাস্ট করবে
ওখান থেকে পাস করা বন্ধ কর বাড়িতে বসে আরামসে ক্লাস করছে চাদরের তলায় বসে অভিরূপ ক্লাস করছে আর এখানে কমেন্ট পাস कौन करेगा
मैम पीके सर को
अरे पीके सर ने क्या बताया तूने क्या सुना
मैम सर नहीं बोला कि एनपी मीट कंडक्ट नहीं करेगा
एनएमसी करेगा
शायद
एनएमसी किया तो भगवान भरोसे है हाँ तो एनएमसी का सिलेबस देखा है वहां पे कुछ ऐसा कुछ खास डिलीशन नहीं है फिर एनएमसी के सिलेबस में व्हाट इज लिविंग दिया हुआ है,
वो न्यू सिलेबस पे एनएमसी नहीं निकाला।
एनएमसी में निकाला है, लेकिन उसमें देखो कुछ कुछ जहाँ पे ना थोड़ा लूपहोल है, क्योंकि वहां पे ना बंद করতে হবে না একটু চলুক তারপরে आई एम जस्ट ट्राइंग टू लूप होल्स पढ़ा के क्या फायदा पे না 5 বছর কন্ট্রাক্ট হয়ে যাচ্ছে
হ্যাঁ এবছরই লাস্ট
তাহলে তো
বন্ধ কোনটা করবে যে গভর্নমেন্ট
আর বন্ধ করতে হবে কি
তাহলে কি বললি কেন
আচ্ছা right. Okay, first of all, if NT is not conducting, NT will focus on NCRT. Now the problem with NMC syllabus is that NMC syllabus they have written ki what is living, when this defining properties have been deleted in CBSE chapter. But what is living you have to study. Then you have to study defining properties. हाँ वो तो पढ़ना पड़ेगा क्योंकि एनएमसी के सिलेबस में व्हाट इज लिविंग बोल के लिखा हुआ है, तो वहां पे तुमको पढ़ना ही पड़ेगा। और एक चीज है एनाटॉमी में वो लोग टिश्यूज हटा दिया, ठीक है, कॉम्प्लेक्स टिश्यू, सिंपल टिश्यू ये सब हटा दिया, फिर भी पढ़ना पड़ेगा। एनसीईआरटी से ही आएगा लेकिन टिश्यू पढ़ाना पड़ेगा, सेकेंडरी ग्रोथ नहीं पढ़ाना है। हाँ नहीं वो तो थोड़ा बहुत पढ़ना पड़ेगा बिकॉज अदरवाइज वोंट बी स्टडी द रेस्ट ऑफ द टिश्यूज ना, मतलब फिजियोलॉजी वाला पार्ट तो नहीं पढ़ पाओगे, समझ पाओगे अगर टिश्यूज ही बंद कर लो। सबसे वियर्डेस्ट था वही कि टिश्यू हटा दिया लेकिन ह्यूमन फिजियोलॉजी लग रहा है, डाइजेस्टिव सिस्टम हटा दिया। The pagal, I don't know what they were thinking. See, this syllabus was made according to because the COVID times. Oh, wife का fight नहीं था।
Syllabus not produced.
बेटे को नहीं मिला होगा। Because it is said कि अभी now it is no, their basic logic was because during COVID so there was a lot of loss of classes and so obviously those who were in COVID times and they were preparing for NEET तो उनका तो फ्लो घाटा हुआ क्लासेस का एंड ऑल। वो भी तो प्रैक्टिकल का भी तो वही तो हुआ था, एक ही तो चीज था। तो इसके लिए एनएमसी सोचा कि चलो थोड़ा रिड्यूस कर देते सिलेबस, इनके लिए अच्छा हो जाएगा। डोंट वरी 2-3 साल के बाद फिर से सिलेबस सेम आ सकता है। देयर इज नो गारंटी टू एनएमसी ना बिकॉज कोविड इज गॉन। आ सकता है, ऐसा तो है, प्रॉब्लम NCRT will change but the essence will remain same. Okay, so that's the thing. Yes, somebody said ma'am. Not ma'am. Somebody said ma'am. Okay, chalo, let's continue. Eukaryotic cells vary in shape and size which determine their functions. We have also studied that they have a well-defined nucleus, they have membrane-bound cell organelles. Okay, so well-defined nucleus present, membrane-bound cell organelles present, membrane-bound cell organelles present. Okay, another non-membranous structure that is seen in animal cells and almost all. Yes. So another non-membranous structure, non-membranous organelle that is seen in central overview তে লিখেছিলাম
না ওটা ইনসেটে লেখা আছে ওভারভিউ অফ সেল।
So centriole is absent in almost all plant cells.
Ma'am.
Plant cell animal cell এর difference আছে না ওইখানে overview তে আছে।
ওভারভিউতে আছে।
Overview তে আছে, that is based on prokaryotic eukaryotic. Here I am talking about animal and plant cell.
Eukaryotic cell
দেখ নিচের দিকে দেখ, নিচে যেখানে প্লান্ট অ্যানিমাল সেলস লিখা ওখানে লাস্ট লাইনটা হচ্ছে অলমোস্ট অল প্লান্ট সেলস
প্ল্যান্টে তো অ্যানাস্ট্রাল রে থাকবে, ওখানে সেন্ট্রাল করতে গেলে
তাই লেখা আছে। হ্যাঁ বুঝেছি বাবা, আমি ওর কনফিউশন দূর করছি। I am clearing out his confusion. overview है prokaryotic and eukaryotic animal cells है yes. Okay, then again you have it here in this. ठीक है तो पाप हीरो को बोलते चलो इसको. Animal cells have centrioles, okay, which are absent in almost all. When you're talking about almost all plant cells, Manya, the reason will be that algae, bryophyte, pterophytes have centrioles. Almost all plant cells means they are minus the higher group, which is angiosperms. Cycas male gamete has centriole because of the multiciliated gamete that it has. Algae, bryophyte, pterophyta. All of them have flagellated gametes except red algae, or they have zoospores. In fungi also you have zoospores, so centrioles will be present. So that is why the statement says almost all. If it was all, that means no plant cell will have centriole. No plant group altogether. So that statement is almost all. If the statement changes, like this is where you go for statement correct, incorrect. If they write instead of almost, they just write all plant cells do not have centriole. That will be a wrong statement. Out and out. Okay, so we say another membranous organelle that is centriole is found in animal cells. Animal cells. Okay, centrioles are absent in almost all plant cells. Absent in almost all plant cells. So, for statement question, remember this phrase. Okay, if I give you this as an assertion, and I say the lower group of plants like algae and bryophytes have centrioles, this will be correct statement.
Yeah.
Okay, so lower group of plants.
Bryophyte, pterophyte.
Algae, bryophyte, pterophyte, all three. Because pterophyte also, the male gametes are called anthrozooids. Whenever you say zoo, that means it has something to do with motility. That means it will be flagellated, biflagellated anthrozooids. Okay, and green algae has 2 to 8 flagella. 8. तो बॉक्स है आज प्लांट किंगडम के वो बॉक्स में दिया हुआ है
Ma'am, you have got a question दिए थे.
हाँ
Some of the C4 plants can place. तो होगा ना.
some of the can respire, all C4 plants can respire. Photorespiration works.
हाँ, photo respiration.
Photorespiration, if we talk about C4 plant, no, they cannot. Because they're this dimorphic chloroplast itself with rubisco present in one, rubisco absent in one. One has granule chloroplast, one is like a granule. What does cam plant do? Cam plant, if you very I don't know, cam plant has been taught. I don't think the NCRT has cam.
हमने किसी लोगों ने अपना डाइट कोड दिए थे, वो भूल गए.
तो তাহলে তুই এই কোশ্চেন আমাকে জিজ্ঞেস করছিস কেন? তখন তুই জানিস ওটা ভুল ছিল।
কিসে বলছি এরকম টাইপের কোশ্চেন আসে
না তো ফটো রেসপিরেশন করবে কি করে? First of all, that there are few pointers you have to understand. Mesophil cell of C4, they have chloroplast, they have granule chloroplast, but stroma does not have rubisco. So that chloroplast of mesophil will release oxygen, but oxygen not getting bound by rubisco because rubisco stroma then nahi. So that oxygen will leave the cell. And here CO2 is being fixed in the cytoplasm. So CO2 is not even entering the stroma of chloroplast. It is getting fixed in the cytoplasm. It is coming to bundle sheath. Bundle sheath, it is getting released. Then it is going to stroma. Now in the stroma bundle sheath, there is no light reaction taking place as such. In the same, like the photolysis will not take. Cyclic photophosphorylation will happen, but the non-cyclic will not happen. Because PS1 is present, PS2 is not present. So that is why their oxygen evolution is not happening. So CO2 will easily bind to rubisco, and rubisco is present in the stroma. So in both those things, one oxygen is getting released but not binding to rubisco, other there is no oxygen being released, but CO2 is binding to this group. So in both ways, it is protecting it from photorespiration. But CAM plants will show. CAM plant shows the same reactions like C4, but in the same cell, mesophyll. Camera day and night. During the day, it is fixing CO2. Sorry, during the night, it is fixing CO2. During the day, it is producing carbohydrates. But it is happening in the mesophyll only. So somewhere or the other, CAM plants might show photorespiration in a very rare case. Suppose the environment is CO2 concentration very low, but oxygen is there because cell is also respiring. So oxygen is there, right? Photosynthesis is happening, oxygen is being produced. But because CO2 is coming in concentration is less and oxygen is increasing in concentration, that point they can show photorespiration. So, okay. At a lower group of plants like algae, अरे কিছু question भाई photosynthesis ছেড়ে দে and pterophytes possess cell sheets. হ্যাঁ সেল পড়াচ্ছি so this is your assertion, that's your reason, option will be 1. A and R are correct, R is the correct explanation, right? So this is another reason. So here we have given one statement, that is assertion. Now NCERT can create a reason for it. Okay, the reason might not be given in the statement, but they can create. They can just add this point here and say ki chalo 10 me now. Okay, whether it's 1, 2, 3, 4, which option. So this is the way eukaryotic cell. Now what we'll do is we'll see the difference between plant and animal cell, but diagrammatically. এবার আকার ক্লাস okay NCERT diagram only I'll focus on drawing. In NCERT Y-shaped ACR is seen in animal cell, which is a little bit wrong. Diagrammatically some wrong things are there, like they have shown lysosomes for both plant and animal. But technically, lysosome is generally found in animal cells. Plant, whatever you call lysosome, is basically spirosome. Microbodies, a type of microbody. They call plant lysosomes. Okay. Diagrammatic difference between plant and animal cells.
Ma'am,
Yes,
please the last part.
just give 1 second. Yeah.
Thank you, ma'am.
Okay, welcome. Online, everybody else has written this part?
Yes, ma'am.
Yes, ma'am.
Okay, very good. So the first we'll do a plant cell. No, side by side, one after the other end. Side by side, there won't be space for labeling. Okay.
ম্যাম আপনার এটার কোনো পিডিএফ আছে ছবিগুলো, তাহলে প্রিন্ট করে বসিয়ে দিতাম।
এনসিআরটি তো ডায়াগ্রাম করছে।
ও yes.
আচ্ছা পিডিএফ পিডিএফ ইজ দেয়ার কিন্তু অন্য ক্লাসের, অন্য আরেক ব্যাচের।
হ্যাঁ তাহলে টাইম বেঁচে যাবে আর কি।
বাবা তুই অনলাইন বসে পাবি স্লো ক্লাস এত এত মানে ডেডিকেটেড স্টুডেন্ট, ভেরি গুড। আমি টাইমটা বাঁচাচ্ছি তোরা। এটা প্রথমবার দেখলাম, আমি জানি স্টুডেন্টস স্ক্রিনশট নেয়, দে টেক স্ক্রিনশট। কি হচ্ছে why this fight? আচ্ছা just wait, let me ঠিক আছে। যাও ওখান থেকে শুধু স্টার্ট রাখতে হয়ে গেল। কোথায় আছে সিআরজি? সিআরজি সেই এতে অনেক দাঁড়া তো এবারে আমি জানি না which one I have to see. Please give those ideas. হ্যাঁ যার আইডিয়া আছে তাকে বল ম্যাডাম। এক্সট্রা পড়িয়েছি ওদেরকে। হ্যাঁ দিস ওয়ান, এইটা দিতে পারি।
হ্যাঁ দিয়ে দিন তাহলে। বাকি
ঘুমিয়ে
না আমি জেগে আছি।
মানে এমন কি রেগুলারলি তুমি বললে না how did it go in hold? Like who's holding my session here? এই ক্যামেরাটাও গেছে নাকি? আর্টিফিশিয়াল ইন্টেলিজেন্স তো আছেই বোর্ডে, কিন্তু খুব স্লো আর্টিফিশিয়াল ইন্টেলিজেন্স। Why haven't I left that meeting? I thought I left that meeting. No, it's there. I don't know what was happening. Okay, so we'll do that. না উপরে তো বডি দেখতে পাচ্ছি seriously I'm struggling more to teach botany than like seriously I struggle. আবার সেই you have no idea how many years the board is studying SAIL. Second. Biopsy, अच्छा secretory membrane বলেছে না মেবি, আচ্ছা এটা প্রোটিন, আচ্ছা ওইটা ওইভাবে গ্লাইওক্সিজোমস আর নিউক্লিয়াস। হ্যাঁ এন্ডোস্টর্ম অফ ইট তো গ্লাইওক্সিজোমস ইনক্লুডেড। গ্লাইওক্সিজোম ওটা ঠিক আছে, বাট ওই secretory membrane, instead of membrane they should have put protein here. তাহলে তো statement would have been correct, but membrane তো there is no secretory membrane as such, there is nothing like that. Protein. Protein. If they would have said कि चलो ER produces proteins that helps in membrane biogenesis, then we can correct that statement. But secretory membrane एक ही yeah. Oh, finally it came. Finally it came. NCERT का mark थोड़ा हुआ है कि you can mark it in NCERT and do the diagram and I'll explain you the differences. Okay. Anyways, I will give you the marked NCERT. I'm almost done with it. Let us finish the cell chapter. Then I give you the entire marked NCERT with where assertion reason is there, where how you. I've also explained certain lines, so everything is there in that. Okay. Chalo. Let us continue.
Plant cell की Golgi body को specially diffusion है.
हाँ.
Plant cell की Golgi body.
हाँ, एकदम उसको डिफ्यूजोम्स ही बोलेंगे बिकॉज उसका अनकनेक्टेड यूनिट्स है.
Golgi body भी तो बोलते.
Golgi body बोलते देखो बिकॉज वो एज सच क्योंकि उसका जो सिस्टर्नल ट्यूब्यूल जो है वो जो सिचुएशन है वो सिक्रेटरी प्रोसेस करवाता है या मॉडिफिकेशन पैकेजिंग करवाता है, इसके लिए उसको गोल्जी बॉडी बोलते हैं. लेकिन उसको डिफ्यूजम इसलिए बोला जाता है कि उसका अनकनेक्टेड यूनिट्स है, उसका कोई सीस एंड ट्रांसफेस नहीं है जो हम लोग एनिमल सेल में देखते हैं. Animal cell will have concentrically arranged cisternae, which will form a cis phase and a trans phase. तो हम लोग जो यूकैरियोटिक सेल पढ़ रहे हैं अभी, it is based on animal cell. प्लांट सेल का यूकैरियोटिक सेल हम लोग नहीं पढ़ रहे. सेल वॉल का जो फीचर है वो सिर्फ सेल वॉल में ही एक्सप्लेन किया हुआ है. प्लाज्मा मेम्ब्रेन का जो कॉन्सेप्ट है वो हम लोग एनिमल सेल के बेसिस पे पढ़ रहे हैं क्योंकि आरबीसी सेल को पढ़ा गया, आरबीसी के मेम्ब्रेन को पढ़ा गया. ठीक है. लाइसोसोमल एक्टिविटी का जो बात करें तो हम लोग एनिमल सेल को ही बोल रहे हैं, सेंट्रियोल एंड एवरीथिंग. ओके. So then we have this another cell wall. This was the middle lamella, and then we have the cell wall structure also. Then we have the plasmodesmata. These depressions that you see in the cell, they are called as pits. Pits has not been mentioned in NCERT. Pits is there in your module. Okay, so pits are the regions or depression where you do not see any kind of cell wall deposition taking place. And through those pits only, plasmodesmatal connections will obviously form. Okay, so coming to internally, they have a large vacuole. Come in. This is the structure, this is a vacuole present, and this vacuole generally is represented as sap vacuole. Okay, so plant cells have sap vacuole. So the structure that is given in NCERT, it is of a mature plant cell. Because immature plant cells like mesomatic cells will not have vacuoles. But when you have young parenchyma cells, they have vacuoles, but very tiny vacuoles. As the parenchyma matures, the vacuoles fuse to form a sap vacuole. Okay, so we have a mature plant cell here. Next, we have a nucleus. Okay, another feature that you have to understand in plant cells, okay, the endoplasmic reticulum is not that as extensive as we see in an animal cell. See the animal cell diagram of here and see the plant cell diagram of here. See the extensiveness. Exactly, because animal cell shows more secretory activity. Okay, secretions are more. So that is why the extensivity of ER is more. Plant cell, you see, it's very like unconnected sort of. Okay, so it's like very scattered sort of. So that's the basic difference here. Structural, I'm talking about. Both have ER, but the structure-wise, it is more complex in an animal cell. Similarly, in Golgi bodies also, it is more complex in animals, and hence we call it as Golgi complex. But in plant cell, we say dictyosomes because it is single unconnected units. They're not connected to each other as such. So that single unconnected units is in module, not in NCERT. Okay. So this is the extent. This is the ER that is present. Is like separated out like that. ACR they have not explained much. They have just given us the ER. Golgi obviously is found. They have shown an Golgi here, but it's like dictyosomes are a single unconnected unit. There's no cis and trans phase there. Okay. Then we have chloroplast. Lots of chloroplasts they have given. Okay. Mitochondria, they have given a few. The structure here is kind of giving us an idea that plant cells, the respiratory activity is not as much as an animal cell. So the amount of mitochondria will also be lesser in plant cells. Okay, and more numerous in animal cell, and depending upon what kind of animal cell that is, then the number of mitochondria differs. Function. Cardiac muscle is the highest because continuously it is beating throughout your life. Okay, so it requires higher number of ATP, striated muscles, liver cells, because too much of production they do. Okay, so many synthesis and everything, detoxification, so many things are happening. So you will have these cells as higher number of mitochondria. Then brain is because now impulse conduction, restoring the resting membrane potential, you require ATP. But brain will have the least when it comes to this cardiac, striated, or liver. Okay. So this is the thing, and next we have SER also. SER like floating in the thing like that. There is no centriole present. There is mitochondria. We have one more structure here, which is called as the peroxisomes. We have peroxisomes, and we are going to study lysosomes also, even though lysosome presence is mostly an animal feature. Okay. The Golgi, ER, nucleus, lysosome, peroxisome, smooth endoplasmic reticulum, rough ER. So let us do rough. So organella and this. So coming to the first point, this is your middle lamella. Middle lamella. This internally green structure is your primary cell wall. Primary cell wall, or we can just write cell wall. Okay. Now here, this is your cell membrane, the red structure. Okay. This is Golgi body, we write, but in technicality, according to module, it is called dictyosomes. Because we are studying plant Golgi, plant cells. So dictyosomes. Then this part is the nucleus. So in plant cells, they have given the extensive description, nuclear membrane, nucleolus, nucleoplasm, and all these things. So we entirely write nucleus. This one is rough ER. Rough ER. Okay. Sap vacuole we have already mentioned. This is mitochondria. This is chloroplast. This is peroxisome. Okay. This is smooth ER. This is smooth ER. And then this is your lysosomes. Plant lysosomes or spirosomes we write actually. And we have the cytoplasm. So one more thing that is missing here is This is called as microtubules. Now, in the NCRT, they have given all these descriptions: sap vacuole, peroxisome, lysosome, cytoplasm, everything. What is the difference from animal cell? Middle lamella is not found. Primary cell wall is not found. Microvilli is not found. So these are the things. Then we have the structure of the ER and all. If you see, both have rough ER, smooth ER, but structurally one is very simple and one is very complex. Okay. Then you have a sap vacuole here, which is not seen in the animal cell. Okay, the number of mitochondria is more. There is no chloroplast, so presence of chloroplast is absent in animal cell. And then microtubules are found in both. But when we talk about cytoskeleton structure, we talk about microfilament and intermediate filaments also. That means plants do not have those two. It is only found in animals. Okay, therefore, maintenance of cell shape is done by what? But that is in plant cell. But you have to maintain the shape of an animal cell where there is no plant cell wall or where there is no cell wall present. How do you maintain the shape? Then cytoskeleton comes into the picture. The integrins, the lamins, the desmins, then actin, myosin, contractile proteins. So these are the ones that maintain the shape along with microtubules. Microtubules are basically cell division and centriole formation. But the rest of the two cytoskeleton helps in maintaining the cell shape. And cell motility also. Cell motility is related to actin myosin because pseudopodia formation is because of actin myosin. Sol-gel theory by Hyman. That was a theory for formation of pseudopodia. So that is by actin myosin. Cell motility. Maintenance of cell shape, those two do. Okay, microtubule cell division. Okay, actin is also used in adherence junction. Calcium adherence, desmosomes. Actin is there. Okay, so they form cell to cell intercellular junctions they form in animal cell. So what you're studying in cytoskeleton is both plant and animal cell. Overall, what we are studying is animal cells. Okay. Only chloroplast, cell wall, and microtubules. These are like we study under plants. Okay. So this is the structure.
Secondary cell wall যেটা বলে
Secondary cell wall basically suppose this is middle lamella. This is middle lamella. Middle lamella দুটো side cell আছে. Here primary cell wall will form. Here primary cell wall will form. Inner to middle lamella. Then secondary cell wall is deposited on top of the primary cell wall. One is inner and one is on top. That is why they say in a mature plant cell, primary cell wall diminishes and the secondary cell wall is deposited. How? Huh? प्लाज्मा मेम्ब्रेन तो उसके अंदर है ना, सेकेंडरी सेल वॉल के अंदर में, एकदम अंदर दो एनवेलप होता है, एक सेल वॉल का होता है, एक सेल मेम्ब्रेन का होता है. तो सेल वॉल में हम लोग प्राइमरी सेकेंडरी को ही सेल वॉल का पार्ट बोलते हैं, मिडिल लैमेला को नहीं बोलते, क्योंकि मिडिल लैमेला कॉमन लेयर है, वो दोनों सेल को सीमेंट कर रहा है, वो सेल वॉल का पार्ट नहीं है. हाँ, सेल वॉल हर एक सेल का सेल वॉल होगा तो प्राइमरी एंड सेकेंडरी सेल वॉल होगा या तो फिर टर्शियरी जहां पे लिग्निन सुबेरिन का डिपोजिशन शुरू हो जाता है. वो जब सेल और ज्यादा मैच्योर होगा ना तब वहां पे लिग्निन सुबेरिन का डिपोजिशन होगा. देयर आल्सो टाइप ऑफ यू कैन से सेकेंडरी वॉल डिपोजिशन. You further deposit lignin suberin, it becomes a tertiary wall. So cell wall can have 3 layers. Primary, secondary, tertiary, depending upon the extent of deposition. हाँ, फंक्शन के ऊपर भी होता है. स्क्लेरेंकाइमा में तुम देखोगे ऑब्लिटरेटेड लूमेन बोलते हैं. ऑब्लिटरेटेड का मतलब क्या है? एब्सेंट ही है ऑलरेडी, क्योंकि इतना डिपोजिशन हुआ होगा, लूमेन ही नहीं दिखता है. तो उसका किस लिए है? बिकॉज टर्शियरी सेल वॉल आ गया है, तो सेल डेड है, वो सपोर्ट कर रहा है. दैट इज हाउ इट इज. चलो, डन फॉर द डे. यस, एनी डाउट्स? नो? क्लियर? चलो, बाय बाय. Please. Anybody doing the diagram? अच्छा, doubt. चलो, online. Bye bye. If you have a doubt, you can ask. হ্যাঁ হচ্ছে বিয়ার আমি একটা আইডিয়া বলবো, তুই ওই কলেজে অ্যাডমিশনটা নিয়ে নেবো।
ওখানে যদি ক্লাস অ্যাটেন্ড না করি
75% বলেছে নাকি অ্যাটেন্ডেন্স?
অ্যাটেন্ডেন্স কত পার্সেন্ট কি বলি জানি না, আইডিয়া জানি না।
আরে এগুলোকে জিজ্ঞেস করে, ফার্স্ট অফ অল লুক এট দ্য কলেজ and see whether মানে ওই কলেজে you see that whether the faculties are good। এবার পাহাড়ের মাঝখানে একটা কোনো কলেজ চলে আসলো যেখানে মর্গে মানে ডেড বডি মানে জানি না কোথা থেকে আসছে, তাহলে তো নো ইউজ টু দ্যাট হসপিটাল। হ্যাঁ বর্ধমান if you get this type of colleges তাহলে তো ভেরি গুড, খুব ভালো, ওখানে ফ্যাকাল্টিও ভালো। না হ্যাঁ কিন্তু I think 2022 তে one of the students they got 617 না 16 পেয়েছিল, কাট অফ ইন্ডিয়া অল ইন্ডিয়া কাট অফ 680, তো মার্কের জন্য পাইনি, বাট হি গট টু বর্ধমান কলেজ। তো বর্ধমান এবার 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 তোর কি তো আবার ইয়ে আছে ঝাড়গ্রাম তো ন্যাশনাল এরিয়া রে, রক্তের হতে হতে আবার ন্যাশনালাইট হয়ে গেলে না না মানে বেসিক্যালি পাচ্ছে। I'm saying you don't miss out that opportunity that you're getting one. ঠিক আছে, you try, you drop for it with that better pass.
But last of the throughout the year, maintain করা খুবই কঠিন ব্যাপার।
না throughout the year meant হ্যাঁ তো you online class তুই আকাশে অনলাইনে জয়েন করবি। আরে when you have the scope to join online, why are you thinking কি আমার হবে না? অনলাইন ক্লাস করবি, ধর ছুটি পেলি, ছুটিতে দিলে আসবি এখানে ক্লাস করবি।
ওয়ার্কে লোয়ার ব্যাচ থেকে ভালো যায়,
মানে ওইটা দুটোই তো করতে হবে। ঠিক আছে, if you want a better college with a better rank, তাহলে I'm not saying you don't miss out on this rank and this opportunity. यानी कि মানে গ্যারান্টি তো নেই, হ্যাঁ everybody leave, those who want to leave please fast. There's no guarantee, you take the admission, you keep it now and you do the class, then you see next year হলে আরো তাহলে চেঞ্জ করে দিবি কলেজ। Yes, sum total of activities and interaction of its constituent cells. Activities. Is of an organism out of sum total of activities and interactions of its constituent cells. So it is a statement it is a statement, right? All organisms are made up of cells and its products. Interactions of constituent cells. বলতে পারি interaction of constituent cells মানে how the cells are interacting, it is like tissue formation, organ system, tissue level. Sum total of activities and interactions of constituent cells. More or less answer কি বলেছে যে কি D কি false বলছে?
False বলছে, all except B. Correct B is false.
B তো false, it's correct only. Constituent of cells মানে unicellular organism কে ধরে নে, তো একটাই সেল সব অ্যাক্টিভিটি করছে, and whatever it is like interacting, it is interacting external, it is because of the products of its own cell. So in multicellular again you have each cell performing its duty but together group of cell forming a tissue. So both ways it is actually explained.
Unicellular
Multicellular দুটো নিয়ে ধরতে হবে, একজনকে ধরলে তো হবে না because unicellular organism না। So even that same question a statement ওদিন কে যেন জিজ্ঞেস করেছিল কি all organisms are made up of cells and its products. I said কি what if I say a statement ki unicellular organisms is a single cell, okay, is made up of single cell and its products. So that is correct only. Yani ki cell theory is for all. on living things, right?
তো ওই যে স্টেটমেন্টটা বলছিল প্লাজমা বেস
Symmetrical in nature. এটা symmetrical তো আবার করে।
ইনকারেক্ট ওটাই হবে।
হ্যাঁ, symmetrical না, because mosaic patterning যখন বলছি, we are not talking about symmetry. Okay. হ্যাঁ,
বলছিলাম যখন রিভিশন করবো
হ্যাঁ।
তো ওটা কি এনসিইআরটি থেকে কোনো নোটস থেকে
দেখ আমার নোটসে তো এনসিইআরটি লাইনই আছে, মানে আই এম নট ইভেন মুভিং হিয়ার দে, 1-2% এক্সট্রা হতে পারে, বাট ইটস ফোকাসিং অন এনসিইআরটি। তো এবার ব্যাপারটা হচ্ছে এনসিইআরটি তে যেটা আছে সেটাকে আমি একটু নোটে এক্সপ্লেন করে বলে দিচ্ছি কি এটা আসাশন, এটা রিজন। তো এনসিইআরটি টা আগে ফোকাস কর, পুরো এনসিইআরটি থেকে পড়, ইউ রেফার টু মাই নোটস ফর হোয়াট এভার এক্সপ্লেনেশন ইজ। ইয়া, মডিউলে না প্রচুর জিনিস আছে, মানে মাথা খারাপ হয়ে যাবে মডিউল পড়ে পড়ে।
ওই আইটিএস এ তো আগের বছর
না না না, এআইটিএস দেখো আকাশ এক্সামের জন্য মডিউল পড়ো। আই এম নট সেইং কি ডোন্ট স্টাডি মডিউল এট অল, তাহলে মডিউল দেওয়ার কি মানে হলো, মানে এক্সামে তো আসবেই। So for Akash exam, you study the module, but I'll tell you what to study because those questions are very common. Polymorphic cell organelle. Okay, gas vacuole is member not does not contain a tonoplast. ওটা কিন্তু কোথাও লেখা নেই NCERT তে, but module এ আছে. So you focus on that, মানে those specific things related to this topic.
কোয়েশ্চেন যখন মডিউলে করার যখন চ্যাপ্টার যখন চলবে তখনই কি এই চ্যাপ্টারটা কোনো শেষ হওয়ার পর
না না একদম
চলবে যখন
চলবে, যতক্ষণে ততক্ষণে তুমি যেন কোয়েশ্চেন তোমার হয়ে যায়। বিকজ কি হবে, সেল চ্যাপ্টার শেষ হবে, উইল বি স্টাডিং সেল সাইকেল, দেন ইউল গিভ অ্যান এক্সাম অন সেল। এবার তুই সেল সাইকেলে ফোকাস করবি না সেলে ফোকাস করবি, সেখানে তোর এই ক্লাসের এই সেল সাইকেলের ক্লাসটা পুরো গেল, ফোকাসটা হারিয়ে যায়।
করছিলাম না কিন্তু করতে যাওয়া প্রচুর খুঁজতে হচ্ছে কোয়েশ্চেন গুলো এবার পরপর
না না, প্রচুর খুঁজতে হচ্ছে, তাই তুই শুধু এক্সারসাইজের কোয়েশ্চেন্স কর না সেল থেকে।
যেগুলো অ্যাসাইনমেন্টের আগে
মাঝখানে মাঝখানে যেগুলো আছে ওইগুলো কর, ওইগুলো করে দেখ। পরে যখন সেল শেষ হবে বা একটা প্লাস্টিক অবধি শেষ হয়ে গেল, দেন ইউ সি দ্য মডিউল কোয়েশ্চেন,
অ্যাসাইনমেন্ট
হ্যাঁ অ্যাসাইনমেন্টস। তো ওইটা ওইভাবে করিস না, এখন তো সবে প্রোক্যারিওটিক হলো, ইউক্যারিওটিক স্টার্ট হলো।
প্রোক্যারিওট যখন পুরো হয়ে গেছে প্রোক্যারিওটের কোয়েশ্চেন গুলো
এক্স্যাক্টলি, হ্যাঁ হ্যাঁ, ওটাই কর, যেখানে পাবি কোয়েশ্চেন ওখানে প্রোক্যারিওটিক পুরো কর।
বাই
বাই বাই বাই বাই অনলাইন।
Bye, ma'am.